pleasure to introduce Dr. Doug Fraser, who is a critical care physician at the Children's Hospital and also a scientist at the Children's Health Research Institute, which is a part of Lawson. Dr. Fraser is also a professor in the Department of Pediatrics at Western University. And during the pandemic, Doug has stepped up to look at the way that the virus interacts with our various body tissues in adults as well as in children. So Doug, can you give us a brief description of the research studies that you have been involved in? I know some have been recently completed and others are ongoing.
Sure. Good afternoon, David. Thanks for chatting with me. Back in March, I think it was March 15th, our first patient came into the intensive care unit here in London. And because of the infrastructure that we have here in London at Lawson and at Western, we were able to start our research studies right away. And we knew it was imperative to get biological samples or blood samples from patients as quickly as possible to have them treated correctly so that they maintain their integrity and to start doing some research on them. We had a few questions in mind, the questions that we thought were the most important to answer quickly for COVID-19. The first question was to determine how the body was responding to the virus and to what degree the immune system was taking off and how that the immune system was causing inflammation. That was the first question we went after. The second question had to do with blood clots. We quickly realized that COVID-19 patients were having serious blood clots, particularly in their lung tissue. So we wanted to determine some of the mechanisms that were underlying these blood clots that were forming and what we could potentially do to stop them or to inhibit them. And the third question we had off the bat was to also identify some biomarkers of disease severity. And that's important for three reasons. We want to know how sick someone is going to become early on in the disease so that we can mobilize our resources quickly, effectively, and from one point of view. And a second point of view, we also want to be able to have very frank, transparent discussions with patients and their family members about the disease process that's taking place. And the final reason is there's going to be a number of large clinical trials coming up. And it's very important to be able to stratify patients into different groups. For example, those that are likely to have worse disease versus those that may have more milder disease, and to stratify those patients early so that we can do the best we can with those clinical trials right off the bat. We don't want to miss anything. So those were the three questions we went after, and we answered all three using blood samples from patients with COVID-19 here in London, and with the infrastructure that's available to us and the wonderful scientists and researchers that are here.
Thanks, Doug. And I think other studies have looked at potential biomarkers as well, but your studies really went much further than that. You really looked at the chain of events as to how the virus interacts with certain cells in the body. And it's not just the lung cells that are involved in, in oxygen transfer. In fact, I think some of your work has highlighted interaction with the blood vessels as being a very early stage in the progression of the intensity of the disease. I mean, can you comment a bit about that?
Sure. So I think if you look at all three of our studies, they're starting to come together into a story, and we're seeing a pattern. And that pattern is as follows. So when you get coronavirus, if you get a big viral load, you get quite an exaggerated immune reaction to protect the body. And that immune reaction involves lots of small signaling molecules called cytokines. You may have heard about this cytokine storm that's occurring. And we were able to, first of all, look at all the different classes of cytokines and determine which ones were really getting elevated quickly in the COVID-19 patients with a big viral load. And we were able to determine that some of those cytokines and enzymes that were elevated were actually starting to attack the host or the person themselves, particularly their lung tissue. Some of those enzymes we've come to realize are also affecting the blood vessels. So the blood vessel inner lining has little hair-like structures called the glycocalyx. And those little hair-like structures keep blood cells like platelets, which are involved with clotting, from sticking to the outer wall of the blood vessel. And what we found in COVID-19 is these same enzymes are cleaving off the glycocalyx. They're literally shearing off the internal lumen of your blood vessels, which is allowing the blood vessels to get inflamed and angry and the platelets to stick. And that's one of the causes going towards the thrombosis or the blood clots. And then along those same lines, when we looked at these biomarkers, we found 6 novel biomarkers, which are all immune related, which accurately predict how someone's going to do once they get COVID-19 with standard care. And those immune molecules that we've discovered, when you look at them a little bit deeper, you realize that they fit into this pattern of what we've seen. So you get a big viral load, you get lots of cellular destruction and lots of debris, and some of those biomarkers are involved with cleaning up debris. So we're starting to see a pattern of things from all of our studies as they come together, which is starting to explain the disease a little bit better.
So does that give us some important clues, Doug, about classes of drugs that you might not normally think would be effective against a respiratory disease? But now you actually know that other tissues like the linings of the blood vessel are actually targets. Are there existing drugs we could repurpose then to get to those early stages of the tissue inflammation?
Absolutely. So we've identified a number of potential therapeutic targets. So if we start again in the beginning with the immune reaction, one molecule, tumor necrosis factor, TNF, typically if you get an infection, TNF goes up very quickly within hours, and then it comes back down to normal levels within hours. And it's considered one of the gatekeepers. It opens up the barn doors to all these other molecules that are being released. What we found in COVID-19 is TNF not only goes up in quite an exaggerated way, but it stays up for the entire ICU stay in the patients. So there's a different pattern that we're seeing with that molecule, and that pattern tells us that it is a potential therapeutic target because it's behaving in a way that we've never seen before. Some of the enzymes that we found belong to certain classes called serine proteases, and we do have effective inhibitors that are safe to give people that can inhibit those serine proteases, and if given at the right time, may be able to stop some of the lung damage that's occurring, as well as the cleaving of those hair-like structures in the blood vessels. So we are coming up with their potential therapeutic targets, and the plan is to move into therapeutic clinical trials in the very near future.
That's great. I think you've also been very shrewd and have taken out some provisional patents on some of the biomarkers that you've seen. And does that, you think, provide a route to translate this knowledge now through the generation of new products in partnership with the private sector?
Absolutely. So part of doing science now is getting involved in the patenting process. And it's very, very important to protect some of the intellectual property so you can go to the bigger pharmaceutical industries, you can go to the larger academic centers, you can go to some of the chemists and say, listen, we've got this intellectual property, we can all benefit from this, but let's work together and let's start moving this stuff forward. What's important with COVID-19 is it came on very quickly. We knew it was a very nasty disease. We didn't know much about it, so we wanted to move quickly. So as we were patenting some of our findings, we made the conscious decision that if that patent process was going to slow down dissemination of the information and getting it out there to other scientists and researchers, we weren't going to do it. But we were fortunate that when we worked with Lawson and Western and World Discoveries, people moved quickly and we were able to go through a patent process, protect some of the intellectual property for future generations and for future collaborations, and at the same time not slow down the process of handing that information out to others so they can benefit from it.
I think this is really important because there's only so much that an individual hospital can do in terms of translation, and this is important information. You want to get out to the rest of the world. So if we look to the next steps, you mentioned there are going to be a number of exciting clinical trials coming along. How can you expand this beyond just London Health Sciences Centre, so you build up a national and international consortium of centres that are going to really, answer these important questions with big cohorts of patients?
Right. It's going to take collaboration, no doubt. Being in London, we saw enough patients and we were able to do good work, but we weren't overwhelmed with patients. So that's a good thing, but it also makes it difficult to do clinical trials. So as you said, we have to create those connections. And we actually do have those connections. We've created them with the Canadian military, for example. They're interested in coming on board in any clinical trials because you can imagine if a large number of military personnel got COVID-19 all at once, that could actually turn into a national threat. So they have a very big vested interest in what we're doing. Because of our patenting position, we've been able to make relationships with big pharma and other companies. And we're looking at alternative funding sources to create those big clinical trials that require large amounts of money because they are going to go across multiple centers. So I think we have that network in place. We've got colleagues down in Louisiana and other places who want to be part of it as well. And as you may recall, Louisiana was a big epicenter, lots of patients. So there's potential to do this, there's will to do this, and there will eventually be the funding and the commitment from everyone to do it.
So I think this sort of research really is a team activity. If you don't have a multidisciplinary team of investigators and health professionals together, this sort of study can never get done. Can you talk a little bit about the number of people and the types of people that needed to come together to form an effective team to get this research happening very, very quickly?
That's actually a really critical point. And it starts right at the beginning when we formed the initial team and we had to go to the ethics department, the biohazards department, and we had the hospital, and we had to say, listen, we want to do this. We need your support to do this. What do we have to do to make this happen? That was the first step. The second step was to go to the healthcare providers, particularly the frontline staff. And if you recall back to March, there were a lot of unknowns. We weren't sure how busy we were going to be. We weren't sure how many people were going to die. We weren't sure if healthcare professionals were going to get it. And so people on the front line were very nervous and trying to get them to then take something extra on by saying, I want to do research now. Can you help me with this? Can you help me acquire these samples? Can you help me with the patients? That's a very, very big ask with everything going on at that point in time. And the frontline staff all stepped up. It was really, really remarkable to walk through the intensive care units, and the nurses, the respiratory therapists, the clerks, everyone had a smile on their face, and they were so happy to be involved in a process where we might find out something about this nasty disease and where we might be able to make a difference. So that was where it moved to. And then once we got the samples, we're doing the type of science that requires a lot of different hands, a lot of different expertise. So while I'm on camera right now, there was at least 10 or 12 people who were authors up front on every single publication who participated. And that includes people in the laboratories, clinicians, statisticians, people who work on artificial intelligence in the mathematical and computer science departments. So it was really a remarkable group of people who came together, and it was all out. People were working every day. They were answering calls at all hours and analyzing data. And if there was a question, they were getting out in the middle of the night and looking at the data and doing things. So it was a full team full team sport right from the beginning all the way through. And that's why we're referring to it as the Lawson COVID-19 study team rather than a few people.
And that's great. It really emphasizes that you have to have a team come together. And yes, only one person can be the first author on a paper, but it is a team, and recognizing that team is crucial, particularly in hospital-based research where no one person can do everything on a study. I think for your studies also, though, it was very important that you yourself went into the ICU every single day and personally collected samples from critically ill patients. And that must have made a big impact and an impression on the frontline care staff to see that your research was absolutely going on there at the front line. And they must have felt really involved in that process and supportive of the research going ahead.
It certainly helps to have a consistent face walking in and saying, I'm here, can you help me? This is why we're doing it. This is what we hope to get out of it. But it was also the appropriate thing to do, because as a critical care physician, I'm used to putting on the protective equipment, taking it off. I'm comfortable working with a level 3 virus. Some of the research associates who would normally collect those samples under my supervision, it wouldn't have been an appropriate thing to do in those early stages. Now we know a lot more. Everybody's comfortable with the process. We know there's not protective equipment. So it's a little bit of it's a different playing field now. But back in the beginning, we were all feeling it out to see how it was going to happen. And that's the way it happened in the clinical areas. And in the lab, it was a little bit different where, the people who were most experienced were working with the samples first, who were comfortable, who knew how to protect themselves and the people around them. So it was a process at multiple levels.
Absolutely. And I think, this research could not have got going so quickly if it hadn't been, with the donors, through London Health Sciences Centre Foundation had not been there and ready to kickstart this research at the quickest possible opportunity. And I think, estate gifts through the foundation were really very important in giving you the means to kickstart this research in a very short period of time. Can you say a little bit about the importance of gifts through the foundation, particularly things like estate gifts that give you the flexibility to really get off the ground quickly?
Right. So, the general public probably isn't used to what we do on a daily basis, where we have to apply for grants. They're competitive grants. You can spend weeks and weeks writing a grant, getting feedback, submitting it, waiting months and months to hear. That'd be a standard funding position we would take in science. But when something like COVID-19 comes on, and it doesn't even have to be that big. It could be something smaller. It could be a great idea. It could be something novel. When you need to move quickly to get something done, you just don't have time to go those traditional routes. So with COVID-19, the writing was on the wall. We were watching what was happening in Italy. We were watching what was happening in England. We knew it was coming. And we went around to all the people who had a hand in this and said, listen, we can do this. We can do it here. We have the ability. We just need a little bit of support. And we started off, as you know, with Lawson and Western, and then we went to individual departments. And then, of course, the London Health Sciences Foundation stepped up with a very big gift. And along the way, because we were getting money and we were making progress early on, then we were able to get competitive funding. So there was a process that took place, and it was absolutely crucial. Without those funds, and the funny part is, it wasn't actually a whole lot of money, relatively speaking. We were pretty good at working on small amounts of money and getting things done. We're pretty effective at that. And people stepped up, provided those funds, and we were able to move quickly. And because of that, I think we answered important questions faster than anyone else in the world.
Yeah, I think it's all about leverage. I mean, a little bit of seed money from Lawson, from the university, then donors coming in, to leverage that money. And then you start to get a momentum, and then you can go to external funding agencies, competitive grant awards, and amplify it still further. But it's got to start somewhere. And the foundation really helps us sort of, get the momentum going on a study, get some initial data that then convinces others, other funding agencies that this is worth investing in. So this is a fantastic example of the value, I think, of giving to research through the LHSC Foundation.
Absolutely.
So thank you, Doug, for sharing your innovation and your insight with us. This is incredibly exciting research. It's ongoing. You made great advances, but I'm sure there's a heck of a lot more to come. And we really look forward to hearing more about it in the future. Thanks very much for your time today.
Thank you. My pleasure.
So now it's a pleasure to welcome Dr. Murat Slezarev to speak with us today. Murat is a physician who works as an intensivist at the ICU at London Health Sciences Centre, and we've invited him to speak about his ongoing clinical trial on inhaled sedatives for COVID-19 patients. But before that, Murat, I wonder if I could ask you just a more general question. So, you were brought up in what was, part of the old Soviet Union, like me, you did your high school training in the UK, you did your medical training in Toronto. What was it made you decide to come and practice at London Health Sciences Centre?
Thank you, David. That's a great question, and it takes me back through my life's journey. It has been an incredible experience to, to live in different countries, to study in different places. Initially, I came to London to do my critical care training here, and at the time, by the time when I was deciding to come here and train in London, I've spent 13 years in Toronto doing my undergrad, doing my master's degree in physiology, then doing my medical school, and then doing my internal medicine training. So we were looking for a program that would be great, and I had a great reception here. I met great mentors in London, Dr. Claudia Martin, who's a co-principal investigator with me on this trial. It was a great experience to come here when I was interviewing, and then I decided to come here for training. After completing my critical care training, I again was mentored by Dr. Martin and Michael Sharp at the North End, and decided to pursue additional training in medical biophysics. I'm actually completing my PhD in that area right now. The thesis has been submitted. I'm waiting for my defense date. But I think the reason for staying in London has been really incredible mentorship and opportunity to do research. This is a phenomenal place to work, clinically, and a very supportive place to do research at Lawson. We also love the city. We have a young family with my wife, so we have 2 girls, 10 and 5. So it was hard to go anywhere else from here. And actually, my wife initially was reluctant to come to London, but when I said, "Do you want to move somewhere else?" she said, "No, we're not leaving. Are you crazy?" So that's how we decided to stay in London, and it's been a great adventure since then. So
that's great. I mean, we're really grateful that you did choose to come to London. It also, I think, shows the value as having top-notch clinician scientists that can be mentors in London that attract people such as yourself. So tell us a little bit about the clinical trial that you're working on with the inhaled sedatives.
Sure. I think it is best if I start with a bit of background and describe why do we need to do this trial, and why do we need to do this trial at this time. So one of the major challenges of COVID-19 is that many patients develop severe forms of pneumonia that require mechanical ventilation in the intensive care unit. And mechanical ventilation is an invasive procedure that involves placement of a plastic breathing tube into the patient's windpipe. This is known as intubation. And in order to tolerate this procedure, patients need to be sedated, which is usually done using intravenous medications. However, the large number of patients that needed ventilation worldwide during the current pandemic and the disruption of supply chains resulted in global shortage of IV agents. And if we run out of these agents, we'll not be able to ventilate patients with COVID, even if enough ventilators are available. You may have seen in the news that there's been a lot of talk about shortage of ventilators. But one of the greatest challenges that we've experienced locally and in Canada in general was that during the peak of the pandemic in spring, there was a shortage of IV sedatives. So we needed to find a solution, and we needed to find a solution quickly. And one of the readily available options are these things called inhaled anesthetics. These are the medications that are used routinely in the operating room to put patients to sleep during surgery. They've been around for a very long time, are safe, inexpensive, and readily available, not just in Canada, but worldwide, especially in places that have low resources. So in addition to that, from previous studies, we knew that they may have beneficial effects on the lungs, including reducing inflammation and opening up small airways, which improves oxygen levels and has been shown to reduce duration of ventilation in non-COVID pneumonia. So the goal of our trial is to determine whether sedation with inhaled anesthetics improves survival in ventilated patients with COVID-19 pneumonia compared to standard IV sedation. We will also establish whether inhaled anesthetics shorten duration of ventilation, whether they can preserve capacity in our ICUs by making more ventilators available, and whether they reduce inflammation and improve neurocognitive outcomes in patients with COVID. We're also doing a study to assess whether use of inhaled anesthetics will actually save money for our healthcare systems because they're cheaper and because they can shorten the duration of time that patients need to remain in the ICU. So hopefully, immediately, this will preserve IV sedative stocks and provide an alternative way to sedate patients in our ICUs.
So that sounds like a real multiple win project, you know, for the health system, for the patient, for scarce medical supplies. That's fantastic. Obviously, you know, the number at the moment, thankfully, of ICU patients is not that high. What's the target population you need, of number of patients you need to recruit to actually get a definitive answer from the trial?
Sure. So because this is a pandemic trial, we aimed for what is known as a pragmatic approach. And because we did not know a priori how many patients we will see in any given region, we needed to ensure that we're doing this across multiple sites. So we're aiming to enroll up to 800 patients across multiple hospitals in Canada. However, depending on how many patients actually are seen during the second wave of this pandemic, we will be able to address the various outcomes in order. So, for example, we've developed something called a hierarchy of outcomes, which means, you know, to answer whether inhaled anesthetics can improve survival, we need the full 800 patients. But with a smaller number, let's say 200 patients, we can answer whether inhaled anesthetics shorten duration of ventilation, which is an important outcome to answer in the context of the pandemic. So hopefully we will reach the full target of 800 patients, and that will be facilitated by the multi-center nature of this trial. So we have centers in Alberta, in Quebec, in other places in Ontario. There are sites interested in United States as well. So we are in the process of expanding to various sites, which is a great feat in itself. So
that's great. So I know the trial is still ongoing. Is there anything you can sort of draw from the early outcomes that would suggest that this actually has a good chance, perhaps, of working out the way that you'd hoped?
Sure. So we actually have not started recruitment yet in the trial, and this is in part because of the novelty of the proposed intervention. In order for us to deliver inhaled anesthetics in the ICU, which is a completely new thing for most ICUs, we need to do three things. We need to get appropriate equipment, we need to create clinical protocols, and we need to educate staff. So unlike IV sedation, which is overseen exclusively by nursing, the inhaled agents will involve collaboration between nursing staff and respiratory therapists with support from our clinical trials pharmacists. So at present, we're working hard at LHSC to finalize those clinical protocols and educate our staff. So we're ready for the second wave of COVID. And obviously, as a trial co-lead, I'm also supporting similar efforts at various other institutions. And we're sort of working against the clock here because we're trying to get ready in time for the second wave. And we're already seeing signals in some parts of GTA and in Quebec that the numbers may be rising. So it's a race against time right now. But again, because of the novelty of this intervention, we cannot, take the education side of this lightly, because it's important that if, in addition, that we do this trial well, we do it safely, both for the patients and the staff. So I can't thank enough the interdisciplinary team in London that's helping me here. There's 2 senior research coordinators, nurses, respiratory therapists, pharmacists, educators, and we had tremendous support from our leadership here in critical care and pharmacy. So everybody's working tirelessly to make sure that this happens in time.
Yeah, I think that's a really, really important point. I mean, firstly, this type of research is a real team activity. No one person can do it. I assume that, in fact, the ICUs at both University Hospital and Victoria Hospital would be able to participate in this study as patients come available.
Correct. So this will be done at both ICUs and medical surgical ICUs and the critical care trauma center at Victoria Hospital.
Thanks. And I mean, you have a lot of both clinical and research experience with patients with pneumonia quite outside of whether they have contracted COVID. I presume this sort of methodology, the use of inhaled sedatives, is really going to be widely applicable to many other types of patients that are going to have respiratory problems quite outside of COVID. Is that correct?
It's absolutely correct. So, you know, the focus, of course, here is on COVID, but we do have a plan to shift to non-COVID hypoxic respiratory failure and other causes of pneumonia, such as influenza, if we don't see a second surge in COVID patients. And it's interesting because, you know, we are aiming to answer the main research question, which is essentially, do inhaled anesthetics improve survival in patients with severe types of pneumonia? We also have a number of sub-studies that we've planned, including assessment of inflammation and inflammatory profiles with Dr. Doug Fraser, looking at cognitive outcomes in these patients. Do inhaled anesthetics improve cognitive outcomes? And this is with Dr. Adrian Owen from the Brain and Mind Institute, and looking obviously at the cost effectiveness of using inhaled anesthetics for sedation. If we show that inhaled anesthetics are non-inferior to IV sedatives, it will provide us with another way to sedate patients in the ICU. And given recent shortages of IV sedatives, this will help preserve our capacity to safely ventilate patients, whether they have pneumonia due to COVID or for other reasons. Now, if we show that inhaled anesthetics are superior to IV sedatives, in other words, that they actually shorten the duration of ventilations in patients with severe forms of pneumonia, this may cause a paradigm shift of how we sedate patients in the ICU worldwide. And they may become the standard of care for similar patients in the future. So this is an interesting intervention for sure, and it may result in some significant changes in the way we practice in the intensive care unit.
So it's we've also had the opportunity to talk to Dr. Doug Fraser. So it's intriguing in that you mentioned that you're able perhaps to get some crossover between the two studies. You know, Dr. Fraser has done a lot of work on the biochemistry of how the virus affects the lung tissue and other tissues, particularly the blood vessels. So you think you'll be able to take some of Dr. Fraser's data and correlate it with the outcome here of you know, inhaled sedatives versus normal standard of care?
100%. And Dr. Fraser and I have been talking. I mean, I was lucky enough to be part of his research team that did some of this early profiling of inflammatory response and to inhaled anesthetic, sorry, to COVID virus. And as part of that work, Dr. Fraser's team was able to identify a set of markers, of protein markers that predict outcomes in ICU. So naturally, our trial offers a great opportunity to validate some of those initial findings. And our plan is to collect blood samples from patients enrolled in this trial and carry out a similar analysis to what's already been done to better understand what happens in patients with COVID, but also to assess the impact of the intervention on the inflammatory profiles. And it will be interesting to see if the anti-inflammatory properties of inhaled anesthetics extend beyond the lung, whether reducing inflammation in the lung, which is the primary location of viral injury, actually it reduces inflammatory damage to other organs such as the kidneys and the heart.
Absolutely. And you know, a center like London Health Sciences Center is so special because we have, you know, experts such as yourself and Dr. Fraser who can actually bring, you know, what start off as separate ideas together, such you have a much more holistic approach to understanding the nature of diseases like COVID. So I know that you've been very successful in getting some external funding towards this, the project for the trial, but you know, I think that the donors have, through LHSC Foundation, have been crucial in actually allowing this work to get going, because you needed some specialist equipment that you simply couldn't obtain through the external granting mechanisms. Can you tell us a little bit about that equipment and why it's so important?
100%. One of the challenges of implementing the inhaled sedation in our ICUs is that we need to purchase specific equipment that could work with our current ICU monitors and ventilators. And out of all of that equipment, one of the crucial equipment, one of the most important equipment is monitors that can carefully monitor and accurately show us the concentrations of inhaled sedatives in patients with exhaled breath. And the goal there is to make sure that we're able to measure concentration of inhaled sedatives in order to titrate it precisely for patient safety. And the monitors are quite expensive, actually, and they do not fall into the budgetary item lines of existing grants that we were able to obtain. So I reached out to the foundation, and they were incredibly generous to support us in purchasing bedside anesthesia monitors that integrate with our existing fleet of ICU monitors. And this will essentially allow us to measure levels of inhaled sedatives and ensure that we can deliver them safely, both during the trial and beyond, if sedation with inhaled anesthetics becomes standard of care. And also, if there are certain indications in critically ill patients where inhaled anesthetics are actually beneficial. Some of them include severe forms of asthma, severe forms of seizures, and difficult to sedate patients who have severe burns or may have resistance to usual sedatives. So in those situations, I think we will be reaching out for those monitors to deliver these medications clinically. So this has been a great support, and I'm really thankful to the donors and to the foundation for supporting this cause.
Absolutely, and it just shows all the stakeholders have to come to the table to get an innovative study like this off the ground. Yes, there's external funding agencies, but our own community is crucial for plugging some of those gaps and actually allowing things to start. So thank you very much, Marit, for explaining your trial and taking the time to speak with us today. We're going to take a short break now, but please continue watching as we're going to share an inspiring video of the importance of legacy giving.
Thank you.
I worked at Union Gas for several years, and that's where I met my wife, Frances. She had an office kitty corner to me, and I thought she was, beautiful, attractive, and I wanted to meet her. I didn't realize at that time that we were going to start seeing each other and eventually get married. When Fran was first diagnosed with MS, it was quite shocking, and at that point we tried to read everything and learn as much as we could. We always had the support of the MS clinic. She unfortunately went into a nursing home only at the age of 50, where she lived for about 6.5 years prior to her passing. We knew the struggles and the battles that we had to go through, and we thought if we can help fund some research, that maybe we can help others in the future. I look forward to the day where they do find a cure for MS, but finding a cure is not cheap, and it requires funding and money. There's great benefits in terms of estate planning. I think it's a smart way for everyone to set up their estate where they can maximize their gift in a tax-saving and strategic way. Sadly, Fran is not with us today, but she fought a hard and courageous battle. Now that she's not here to fight, I am, and I'll continue fighting it in her memory and her legacy.
So it's now my pleasure to introduce Dr. Tarek Lubani, who's an emergency room physician at London Health Sciences Centre and associate scientist at Lawson. Tarek has a long history of creating low-cost medical devices for use in parts of the world where healthcare supplies are less abundant than they are in Canada. And he's been awarded international prizes for this work. But during the pandemic, he has applied his innovation skills much closer to home. Tarek is joining us to discuss the creation of a novel, non-invasive ventilation mask that could help in preventing patients from having to move to ventilation, which involves a tube down the throat and all of the associated problems like heavy sedation. So Tarek, can you give us an overview of the research that you've been performing and really start with, where did the idea for the mask come from?
One of the things that we've known for quite a while is that the non-invasive techniques of ventilation, that is the ones that don't involve putting a patient into an induced coma, are techniques that are dangerous for the people around the patient. So we have a mask that we put on people's faces and then we start blowing high pressure air without really a very good seal around it. And that means that it's dangerous to other patients and it's dangerous to the healthcare providers who are around. I was taught this in medical school years and years ago, but so we've known it, it's not a new problem, but this problem wasn't something that we felt we really needed to do something about, because ultimately the use of BiPAP wasn't all that pervasive and the risk of infection was acceptable. We sort of thought, okay, so my patient gives me the flu. That's a hazard of working in the emergency or working in some of these care centers. And it's worth it because I pay the cost and my patient gets the benefit. Non-invasive ventilation is less dangerous for the patient, more likely to get them out of the hospital sooner and healthier. So when we hit COVID, finally the equation didn't add up. It didn't make sense to expose healthcare providers and other patients to these aerosols that are being generated like crazy, at the rate of 100 liters per minute. We're generating aerosols when we're using BiPAP with regular masks. And that was the genesis of this idea. Now, this idea hasn't really been tried that much before. And so we knew kind of how it would work. Other people had started working on it during COVID, but we didn't know exactly how it was that we were going to make it happen. And that's where we started looking to other industries, like, for example, who else needs nothing to get into their mask and nothing to really get out? Firefighters. And so we took a firefighter mask, started modifying it, and turned it into something that we could provide oxygen to our patients with without anything happening to the people around them, without other people being exposed. That's
great. So did you literally just start with a firefighter's mask off the shelf sort of thing and then adapt it, or have you had to do some materials modifications for use in a clinical environment?
For us, it was very important to be able to take this technology that we worked on and developed and to be able to apply it really anywhere, to be able to give it to anybody who needs it anywhere in the world. There are firefighters pretty much anywhere where there are hospitals, and they generally have masks. This is more or less universal. It was not just our team. We were in collaboration with a team from General Dynamics who have lots of experience with these kinds of things. And all we wanted as LHSC, as Lawson, was to basically say to people that we need you to the designers, we need you to give us something that we can use anywhere and that's cheap. And that meant that we had to start with something that already existed. to create it from scratch and tool from scratch, especially in the midst of a global pandemic, not only would have been difficult, it would have been impossible. And so not only did it make sense in terms of reducing the cost and reducing the production time, it also made sense just in terms of the resources available at a time like this.
Yeah, and I guess having a partner like General Dynamics is really important because, in their line of business, they have a lot of expert knowledge about protective equipment. And I guess that was the ideal partner, really, to go into this sort of endeavor with.
They were a really great partner. They, I think, had the engineering chops to make something like this happen. And with their engineering chops and with LHSC and Lawson's sort of research chops, we knew that we could take on a problem that other people had been thinking about, other people had sort of started working on, and actually beat the problem back.
So how is the research actually coming along? I know you probably need, quite a sizable cohort of patients to really, show the utility of this new design. Where are you in the process at the moment?
Well, if you don't mind me sort of showing the mask a little bit. So
Please
basically,
do, yeah.
Yes, this is the mask. And essentially this part here is the firefighter mask. This is a 3D printed part that converts it from a regular firefighter mask to something that we can put on our medical devices. And then we added some safety features like the strap, which allow the very safe and quick ejection of the mask if anything is going wrong. So when we got this done and figured out the production, we've also open sourced it, which I think we'll talk about in a moment. Then we started turning towards getting the ethics straight and starting to test it on patients. We have a seasonality to the use of BiPAP. And of course, very luckily in London, we didn't have a very large first wave of COVID. And so we started testing it and got a really good idea of how this mask worked and got some preliminary data after the first few months. We're about halfway through our data collection right now, which is great. And there has been a lull through the summer of BiPAP use because not as many people need it. There's always a lull every summer. So we're now starting to pick up. Actually, we just ran a patient last night, and we expect that we'll be able to get the rest of the patients sorted out over the next few months.
Well, that's really encouraging. And while it was COVID that really gave the impetus to move this idea along, I guess this is a piece of apparatus that would really be applicable for other patients with any sort of risk of an infectious disease that would protect them and also protect the healthcare workers.
By definition, the patients who are using this mask are very sick. You wouldn't put this on somebody who isn't. And infection is both in terms of overt infections, I can tell this person has a pneumonia, or covert infections, superimposed or hidden or occult. These things are such a part of BiPAP that we've known this is a problem. And I think it's probably high time that we stop putting our respiratory therapists, our nurses, and our other patients at risk. Even after, you're right, COVID was the impetus, and I think COVID is probably one of the most important uses here. But even after that is done, these masks will be usable by so many patients in so many places so that they can reduce the risk to themselves, to the people around them, and to the healthcare teams. I think it's something that we've owed to the healthcare teams for quite a while. And I'm glad that we're finally able to realize the promise of safety to the people taking care of our most vulnerable patients.
Which brings us on then to how are you going to, distribute this knowledge and make the mask available? And you mentioned, you want to make it open source. Can you sort of just describe the translational plan to get this mask, into wide use within, not just within Canada, but potentially, around the world? And what's your business strategy for getting that distributed?
We have a history now of making other medical devices that are very expensive on the market, but very cheap to produce. And the reason why they tend to be very expensive is that maybe there's a lack of knowledge in how to produce them in a high quality way. So for example, our group started with something very, very simple about 6 years ago now, the stethoscope. I have one here with me actually. So this is a stethoscope that's 3D printed. 3D printer doesn't have to cost much. It's only a few $100s, and then uses literally the tube from Coca-Cola fountain machines, and basically allows us to create a stethoscope that sells on the market for $200 to $300 for $2 to $3. $2.81 US actually was our breakdown. So we know that putting things out there makes them available to patients who might not necessarily be able to get them otherwise, and healthcare providers who might not necessarily be able to afford them otherwise. In London, would it change things if this mask cost $500 or $5,000? Probably not. But there are remote parts of Ontario where healthcare providers don't have that kind of funds, where healthcare providers aren't using high quality medical devices because they're not available. And they're not available either because they're too expensive, or they're not available because they just can't get them in. Right now, at any cost, you wouldn't be able to get these masks in. That's why what we do is we take the entire data set. So not just the research data, that's important, so that people can look at our data and verify it, but also how to produce the mask. So all of the plans are already available online, and all of the processes, how you go from having these parts for this mask to actually having a cohesive working mask. And then even more important than that, how you can check that the mask works.
That's great. So I guess from a patient point of view, this is so reassuring because, you know, I've heard anecdotes of from ICU physicians that when they tell a patient they're going to have to go onto mechanical ventilation, and you know they know they're going to be heavily sedated, they know that the recovery from that is going to be long, and this option must be so welcoming from a patient point of view.
One of the things that's happened very early on was a lot of patients with COVID were put immediately onto invasive mechanical ventilation, that is to say induced coma plus a tube all the way in. Lots of them did very, very badly, and that was for a number of reasons. But one of the things is that we were taking patients who had one problem and we were introducing another one. We early on had this bargain. We thought, okay, this is the cost is worth it. What we've learned over time is that COVID patients especially, but some other patients as well, they have a problem, but it's not a problem that needs us to, as it were, overreact right away. When we reduce the risks to a patient, we also reduce the bar for use. For example, if I have a patient who's elderly and not likely to survive mechanical ventilation, I'm going to be reluctant to start them on mechanical ventilation. So I'll wait until they're sicker. And of course, the longer you wait to start putting out the fire, the bigger the fire. With these relatively safe, non-invasive options, not just safe to the patient, but safe to me as well and my healthcare team, we can start sooner. Lots of hospitals all over Canada and all over the world banned non-invasive ventilation because it was so dangerous to other patients and to other healthcare staff. And this puts it back in their arsenal so that they can use it and keep their patients and themselves safe. Would it necessarily change outcomes in London? In London, we have negative pressure rooms. Maybe not. But in lots of places, for example, we're working with a remote region in Sioux Lookout, they don't have negative pressure rooms. And non-invasive ventilation was not an option. They would sit there hours and hours away from the nearest mechanical ventilator, watching their patients deteriorate while they waited for transport. And that's a tragic situation that this device will avoid.
Absolutely. You know, getting a study like this started, speed was of the essence, because I mean there was a need. You had the patients did not have a solution. Your idea could provide a solution, but you had to build the prototypes very, very quickly. How important was the opportunity of having you know, donors through the LHSC Foundation be ready to almost like crowdfund this, to really get it off the ground quickly. Could you have done it without this sort of access to funds?
To ask that question is like asking, can a team operate without one of its vital organs? Donors are, I think sometimes donors might be treated or are thought of like wallets. They're not, they're team members. They're really vital organs. And the way that we looked at it when we started talking to some of the donors who worked on this project is we had different skill sets and different abilities. We were able to provide different things, but we all had the same goal, to make people healthier and to make the world around us safer and better. And when we had that goal, the main question wasn't for us, what are we going to do about this? Or for them, what are they going to do about this? You know, we're scientists, we're going to do science. And the donors are able to provide some of the finances. But it was really, how can we work together as these different vital organs to make sure that the key project, the key goal can be met? And so really, it's hard to say because it could sound tokenistic to say, oh yeah, we couldn't have done it without donors. But really, I think the way that I contextualize it is that we're all these vital organs in one key body of science, and it would have never worked without any one of those vital organs.
Yeah, that's a great point. I mean, there's so many different partners. There's the knowledge and skill of people like yourself. There's the engineers. There's the private sector partnership with companies like General Dynamics. And then there's the donors. And it's only when they all come together that smart ideas actually become products, and products improve, you know, the health of our patients. So there's a great opportunity to show the real value of donors being partners in this innovation system. Finally, then, how are you going to handle distribution now? So you've sort of got proof of concept. Yes, you're still gathering data, but in terms of now distribution, you're very much looking to provide the instructions for different centers to actually now build their own, rather than actually make a product and distribute the product. So in terms of backup, how are you going to help various centers around the world, to actually make sure that they can not only construct, but the quality control is there on the device as they build it in their own centers?
We've done something really interesting with this project that hadn't been done before, which is to take these devices. Some other people have published how to make devices, but we've taken these devices and we've put them through regulatory requirements. So for example, this device is Health Canada approved, and we're just about to complete our FDA approval as well as a Class 2 device. So we prove to ourselves and to others that this device is usable in the first world, on the world's best cared for in a regulatory standpoint patients. Once we do that, then basically the same project, the GLIA project, which operates out of LHSC in the Division of Emergency Medicine, and also sort of operates as a Health Canada approved and licensed facility, that group of people will make also these devices and make them available for purchase essentially at cost. And so anybody who wants to use it or buy it can do that. You can sort of think of it like a recipe book. You know, you might have a recipe book, you might know exactly how to make the finest spaghetti or pizza, but you might not want to make it yourself for whatever reason. So we provide the product for people. As well, we provide the recipe book. You know, some people are not within our purview or don't want necessarily for us to be making these devices for them. So we work with ministries of health, we consult with them, and we give them any information or expertise. For example, in Tunisia, there was a project where they wanted to create some of these devices. And so we sent some of our engineers over there to work with their engineers to get a production line up and running. And we've done the same in other parts of the world wherever really requested. And the third, of course, we are first and foremost scientists. And so we publish in scientific journals so that we can also put the rigors of science to these devices, and so that we can open ourselves up. Our goal is not to create a device and put it out there. Our goal is to make people's lives better and make people healthier. And a lot of times when we make devices, these devices are going to need improvement. And so what we do when we translate that knowledge out into the world through publications and through social media and such, is we invite modifications. My favorite email to receive is, I like your idea, but, and then there comes a really beautiful sort of next piece for it, that in which somebody suggests an improvement. And often we will say to them, thank you, that's a really great idea, we'll implement it, but we need your help. And we create a collaboration on the spot. And that's happened with this project, that's happened with other projects. And it's one of the best things about science is the open and collaborative spirit.
Very much. Constructive feedback is the best thing you can ever receive, I think.
Yes.
So thank you very much for taking time to talk with us today, Tarek. We aspire to innovate for the world at London Health Sciences Centre, and I think this is a great example of exactly how we can do that. Thank you very much. Bye-bye.
Thank you so much, David.